All right, and we're going to finish up with our RNA viruses. Last time we ended up right here talking about enveloped, positive sense, single-stranded RNA with the Coronaviridae family, or the coronaviruses. We're going to start today with our envelope, positive sense, single-stranded RNA. But big difference between these two is that this is a group of viruses that contain an enzyme called reverse transcriptase, which means they can do reverse transcription. Now, the families of viruses in this group, it's only one big family of a couple different viruses. It's called the Retroviridae group. So retro means backwards or back, and so they all do transcription backwards. Now, of the retroviruses that are in the Retroviridae group, the genomes, they all have two identical strands of positive sense single-stranded RNA. So they are segmented RNA. They have two identical strands. So they don't just have one copy, they've got two. And they can transcribe DNA from RNA. So they can make DNA from RNA. And they do that using the enzyme called reverse transcriptase. Now, viruses in this group are going to cause one of two issues. Neither of them are good. The first issue is they can be oncogenic, which means that they can generate cancer or they are cancer-causing. The next group is called immunosuppressive, which means, as the name implies, they are going to suppress the immune system, and that's where the HIV comes from. So, how do they do transcription in reverse? So here's that single strand of positive sense RNA. They're going to use an enzyme called reverse transcriptase, and they are going to read it, the A's, the U's, because it's RNA, the C's and the G's, and they're going to bring in the DNA bases, the A's, T's, C's, and G's, and make a copy. So they're making that first copy. They're then going to take that single strand of DNA and get rid of the RNA, don't need it anymore, and they're going to take that one strand of DNA, bring in the opposite bases. If there was an A, they'd bring in a T. If there was a C, they'd bring in a G. And by the end, they have double-stranded DNA. So we went from RNA to double-stranded DNA. Now, the first group of retroviruses that we're going to talk about are the cancer-causing ones, the oncogenic ones. Now, the two main viruses in this group, the one is called the human T lymphotrophic virus, and the other one's called the human T lymphotrophic virus 2. The human T lymphotrophic virus 1 causes cancer of the white blood cells. So it's adult acute T cell lymphocytic leukemia. Now, anytime you have cancer, again, cancer means you have cells that are reproducing uncontrolled. And when they do that, they can't check themselves for abnormalities. So a lot of times they're going to make incorrect cells. So we make large quantities of incorrect cells that pretty much are pointless. They can't do anything. They can't do whatever they were supposed to do. The human T lymphotrophic 2 causes a condition. It's still a white blood cell. It's still T cells. It's still part of T lymphotrophic. It just causes a little bit of difference in the shape of the cells. They're called hairy cells. I don't know if I say they're hairy, but they do have these extensions of the white blood cells that stick out, and it's where they get the nickname the hairy cell leukemia. Now, both of them, the virus can get transmitted by blood transfusions, using contaminated needles, sexual intercourse, and even breast milk. Now, an issue is that we don't have any treatment. We could try to do various types of cancer treatments, but none of them have really been all that great at getting rid of the underlying cause. The fact that this is a virus that's causing cancer. We have lots of things that can cause cancer, and there's lots of things that can trigger cancer, but the virally caused cancers, specifically these, have been really hard to treat. So they usually cause a chronic condition of your white blood cells, and the long-term prognosis is poor. Your best bet is prevention, monogamy, no needle sharing, condoms will help, all of those just to prevent yourself from picking up of one of these particular virus strains. Now, the other group of retroviruses are the immunosuppressive. So as their name implies, they're going to suppress the immune system, so the immune system can't do what it's supposed to do. Now, if your immune system is no longer working, like at all, you develop a condition known as acquired immunodeficiency syndrome. You acquired it. You weren't born with this immunodeficiency. And so it's an acquired immunodeficiency syndrome. Now, things that happen when you're in this AIDS stage of HIV infection, one, you're generally going to have opportunistic or rare infections that healthy individuals would not normally see. Two, you're going to show antibodies for HIV. Your immune system has recognized that it is infected with a virus, and it can make antibodies against it. Not that that's going to cure you. And your CD4, your T helper cell counts, are going to be below 200 cells per microliter of blood. Normal counts are between 400 and 1600. So 200 is way below normal. Now, the human immunodeficiency virus, HIV, is the virus that causes acquired immunodeficiency syndrome. This virus destroys, ultimately, the immune system. Now, the human immunodeficiency virus only replicates in humans, so that's a good news. But the question then is, well, where did it come from? Because HIV hasn't been around forever. We first started seeing cases of it in the United States in the 70s. And although there was cases of it prior to that, it hasn't been around for hundreds of years. So right now the resounding theory is that it came from the simian immunodeficiency virus. Now, simian means monkey. So monkeys have their own strand of immunodeficiency virus. And for some reason, that virus changed just a little bit. And again, RNA viruses are known for doing that. So it changed just enough that the virus can now infect humans. Now, it destroys the human immune system, and there are two strains of HIV. They both cause the exact same thing, but genetically they are different from each other. HIV-1 is found more in the United States and Europe. HIV-2 is found more in West Africa. Now, I'm going to make note of this little diagram that I have over here. This is the basic structure of HIV. The inside of it, the genetic material, it's got two identical strands of that single-stranded RNA, and the outside of it has two different glycoproteins, glycoprotein 41, glycoprotein 120. These glycoproteins are used to recognize our immune system cells and target them. So it can't go and attach to any other random cell in the body. It uses these very specific proteins so that it attaches to our T helper cells. Now, it uses those proteins to attach to our T helper cell, our CD4 cells. Then, and I'm going to hopefully get this video to work, and we're going to go through these steps with the video. It enters into our T helper cells by endocytosis. So the whole cell goes up and engulfs pretty much the entire thing, the envelope and everything. Well, then it's going to uncoat. It's going to get rid of everything it doesn't need. It's going to get rid of the envelope. It's going to get rid of the capsid, and now you have your single strand of RNA. Then we have to make DNA. So we're going to use that reverse transcription, and we're going to make our double-stranded DNA. That DNA is going to go into the nucleus, because that's where our DNA is, and it's going to incorporate itself into our DNA. So it inserts itself right into our DNA. Now, that means your DNA has viral DNA in it, and your DNA has now been hijacked. Your cells have the DNA on how to make more viruses. And so we then start to get into the synthesis of RNA. We're going to start to make some of that viral single-stranded RNA through transcription and make all the proteins that go along with it. We're going to make all the parts. So we're going to do transcription, but making viral RNA, and we're going to do translation, making viral proteins. Then we will have to assemble all of them, put them together, and they get released. Now, a lot of times they're not fully assembled when they get released. After they get released, then everything truly comes together, and it will go off and find new cells to infect. Now let's see if I can get my video to work.
When HIV first enters the human bloodstream, the virus circulates throughout the body. The protective surface of HIV is studded with particular proteins called GP120, and inside is a capsid that contains the viral RNA and viral enzymes. Initially, the GP120 protein on the virus is only able to bind to a cell surface protein on the macrophage called the CD4 receptor. A second receptor protein, CCR5, must also be present on the macrophage for the virus to enter the cell by endocytosis. Viral RNA and enzymes are released into the cell's cytoplasm. Reverse transcriptase uses the viral RNA to synthesize first a strand of viral DNA and then the complementary DNA strand. The viral DNA then moves to the nucleus of the cell where it integrates into the host cell's DNA. Transcription of the DNA within the nucleus now results in the production of viral RNA. This viral RNA can serve as the genome for new viruses and can be used as messenger RNA to produce viral proteins during translation by the ribosomes. Complete viruses are assembled and released from the cell via exocytosis, which causes little harm to the macrophage. HIV cycles through macrophages over a period of years and continues to multiply while doing little apparent damage to the body. Eventually, the gene that codes for the GP120 protein is altered by mutation. The altered GP120 protein changes its co-receptor allegiance and now binds to a different co-receptor, CXCR4, which is found on the surface of CD4 plus T cells. The same processes occur within the T cells, resulting in new virus particles. However, as these new viruses leave the T cell, they rupture the plasma membrane, which kills the cell. As these newly released viruses invade and destroy other T cells, the body's immune response is weakened, and this leads directly to the onset of AIDS.
Now, the course of HIV eventually leading to the AIDS stage. The primary infection. So we're going to look at HIV in the blood. There's not a lot right away, but it does skyrocket within the first few weeks. Your cells do become virus production factories. However, and this is the same time a lot of your T cells are dying, but you are making antibodies against it. So between the antibodies increasing, we do start to get rid of some of the virus in the body. However, your body is not equipped to get rid of all of the virus in the body. So we always have this big spike in viruses right away, and this is usually when we see some of the initial symptoms, although not everyone has symptoms, some of the initial symptoms of an infection. Then they subside. Then we jump from weeks to years. The amount of HIV in the body can remain relatively low. This is when we say a patient is in clinical latency. It's gone dormant. It's not doing a lot in the body that we can visually see. However, it is still a slow progression of your T cell count. Now, when your T cell count becomes low enough, when we get down below that 200 microliters, or 200 cells per microliter, we now say that you've gotten into the AIDS stage of HIV infection. You're going to start to develop opportunistic diseases that people that have healthy immune systems develop, and it's those opportunistic diseases that eventually lead to death. The HIV doesn't kill you. It's the fact that it kills your immune system or destroys your immune system. You're going to develop some disease that you can't get rid of because you don't have a working immune system. Now, a couple diseases that are associated just with those that are in AIDS stage of HIV infection. This picture on the left is actually a disseminated herpes virus. This is herpes 1 or 2. It could be either one. And again, you with a healthy immune system might get the occasional lesion that will go away. In an AIDS stage patient, these lesions aren't going to go away. They're going to continue to spread. The picture on the right, you might recognize it from another PowerPoint when we talked about viruses that can cause cancer. This would be the Kaposi sarcoma caused by the human herpes virus 8. It's the cancer of the blood vessels. So AIDS can cause some serious issues. Again, things that healthy people would not pick up. Now, the spread of HIV is that it was first discovered in young male homosexuals in the 1980s in the United States. Again, there were cases of that before that in the world, but this particular virus is worldwide. Every country has HIV. It's found in various bodily fluids. We can find HIV viruses in blood, semen, saliva, vaginal secretions, breast milk, of all of the various bodily fluids that you could find the virus. Blood and semen are going to be the highest concentration of viruses, so more infectious, and breast milk is up there as well. Now, I always get the question of, there's viruses in saliva? There are. However, with any other organism, you need to hit a certain infectious dose to actually pick up the disease. Well, because the virus is in such low quantity in saliva, you'd have to drink a gallon of saliva to get the infectious dose for this particular virus. So I don't think anyone's going around drinking a gallon of saliva, so we consider it a non-infectious fluid. Now, this is just showing where we have the highest numbers of cases of HIV worldwide. And yep, here in the United States, we're top. Otherwise, lots of areas of Asia and down in Africa, the southern parts of Africa, again, they have their own strain of HIV, but it still causes the same issue. HIV transmission, it actually, because it's commonly found in both semen and blood, depending on whether it's an adult male or adult female, actually kind of breaks down differently based on how it's spread. So in males, the top way it's spread is male homosexual contact, followed by high-risk heterosexual contact based on various types of injected, like needles, drugs. Females, top way it's spread, high-risk heterosexual contact, then followed by injected needles or drugs. Now, diagnosing someone that's in the AIDS stage, again, they're going to look for symptoms. Some of the initial symptoms is unexplained weight loss, fatigue, fever. Sometimes you almost get this white kind of shade or coloring to it in the mouth, but it can vary among a lot of individuals. As you further progress, you're going to see some of those rare opportunistic infections. You're going to have a low white blood cell count, less than 200 per microliter of blood, but they're also going to do a diagnosis looking for antibodies against HIV. And the two main ways they do that is either an ELISA or a Western blot test. Now, as I already said, one of the main ways they test is looking for antibodies, an ELISA or a Western blot. Now, just because you have antibodies doesn't confirm that you have HIV. You may have been exposed to a non-infectious dose and your body recognized the virus, even though it wasn't enough to cause disease, and you develop antibodies against it. So even if you have antibodies, it doesn't mean you're not going to be able to get the virus in the future. You can't make enough antibodies, but it is a clue. It's the first clue, because we can run a lot of tests on an ELISA or a Western blot. So it's the first clue that someone may have HIV. If you are showing presence of antibodies for the virus, then they'll do a PCR test. They're going to look for the RNA for this particular virus. Otherwise, they're going to look, they're going to try to look at signs and symptoms, but because it can vary so much between patients, it's not the main way they do a confirmative diagnosis. They're going to do a PCR. Now, the interesting part is some don't develop AIDS or HIV infections. Even though they've been given an infectious dose, there are sometimes that the virus can somehow become mutated or defective. Sometimes they can't bind to our cells correctly. And there are a few cases where people truly do have a strong enough immune system to get rid of it. Extremely rare, and they make the news and usually medical journals when we find someone that has an immune system that can literally have immunity to this virus. Now, treatment, they call it an ART, which stands for antiretroviral therapy. Means you're going to take at least 3 antiviral drugs from at least 2 different viral drug classes. So we've got lots of different types of viral drug classes. Just like antibiotics, we put them into different classes based on how they work. Well, we've got lots of viral drugs too that we classify based on how they work. And so you're always given at least 3 drugs from at least 2 different antiviral drug classes. All of them are to decrease viral replication and release. so that that virus stays under control and pretty much keeping it dormant. Now, the best bet is to not pick up this virus to begin with, so prevention is all behavioral changes. So safe sex using condoms, clean needles, universal precautions, especially in the healthcare setting, making sure you've got gloves, eyewear, masks, screening blood products before giving them so that we're not spreading it. Now, I put my little line. So we finished. These were all positive strand, single-stranded RNA, and then this one even had the reverse transcriptase. We're going to finally move on to our next section on here, all of our negative single-stranded RNA, so the ones that the ribosomes can't recognize and read. Now, our first group that's enveloped, but these are all going to have one big, huge, long single strand of RNA, so non-segmented. So it's one big, long strand of RNA. Now, there are three families. There's the Paramyxoviridae family, which is our biggest one, the Rhabdiviridae family, and the Filoviridae family. Now, anything in the Paramyxoviridae family, all of the viruses will always form a syncytia. This is when infected cells will all fuse together. They do that to help evade our immune system. They can easily sneak between cells, between areas, and our immune system can't always recognize them when they're all fused together. Now, there are four groups of viruses in this family. There's the measles, the parainfluenza, also called the paramyxovirus, the mumps virus, and the pneumovirus, also called the respiratory syncytial virus. It even has that syncytia in its name. Then we're going to go through the Rhabdiviridae family, which causes rabies, and the Filoviridae family, which has a few emerging diseases like Ebola, as well as a few others. So, starting with our first family, the Paramyxoviridae family, and our first virus in this family, it's the measles virus. Now, it's spread usually in large, dense populations by respiratory droplets, and the virus gets into the body through the respiratory tract and it spreads throughout the body. Now, one of the diagnostic tools and symptoms are what are called Koplik spots. There are these white spots that always happen in the mouth, and they usually have kind of a little red inflamed halo around it. Now, other than having the Koplik spots in the mouth, you usually end up having almost a rash or lesions on the head that goes throughout your entire body, as well as fever, sore throat, headache, might develop pink eye or coughing, and that usually all happens in about 7 to 14 days after infection. Now, most of the time from there, you can recover. However, this particular virus can lead to some very rare complications. It can lead to pneumonia, encephalitis, and something called subacute sclerosing panencephalitis, which is hardening of the brain tissue. Now, the last big outbreak until your textbook came out there was a big outbreak of measles at Disneyland, hence my little picture on here. So, however, as we've seen just in the last year to 2 years, we've had lots of measles cases here in the United States. And it's usually found in areas where there's a high percentage of people not vaccinating for this particular virus. Now, diagnosing for measles, a lot of times they're just going to look for the signs and symptoms, including those Koplik spots. Treatment, if they can, they usually try to give antibodies within 6 days of exposure. They can also give vitamin A injections. That seems to reduce the seriousness of the symptoms. Otherwise, best prevention is we have a vaccine. It's the MMR vaccine. It's the M of measles, mumps, and rubella vaccine. It's pretty effective, and when enough people get vaccinated, we can really see the big decrease in number of cases of measles. However, we are starting to see that uptick in the number of measles cases every year. Now, our next virus that's in this Paramyxoviridae family is the Parainfluenza virus, also called the Paramyxoviruses, and there are 4 strains called the human parainfluenza virus 1, 2, 3, and 4. Now, children and infants are always going to be at highest risk of picking up this particular virus that was transmitted by respiratory droplets, person-to-person contact. The human parainfluenza virus 1, 2, and 3 all affect the lower respiratory tract, whereas the human parainfluenza virus 4 affects the upper respiratory tract. Now, they're all going to cause inflammation of the respiratory tract. They're going to have basic symptoms of respiratory tract infection, usually coughing, sometimes runny nose, sometimes just more difficulty breathing. But the human parainfluenza virus 1 and 2 can cause a condition known as croup. It's inflammation of the larynx, the trachea, and the bronchi. So when the coughing happens, it has a very unique sounding cough. It's called a seal bark cough. If you've ever been to a zoo and heard a seal make its noise, its barking cough, that's what this particular coughing sounds like. And it's all because of the inflammation of these parts of the respiratory tract. See if I can get the sound. So it's a really deep kind of barking sound. I always can literally picture in my head a seal making these noises as they talk to each other. Now, we don't have any treatment for the parainfluenza virus. Luckily, most recover within days without any serious long-lasting issues. I don't want that again. Now, the third of the Paramyxoviridae family is the Mumps virus, also known as the rubella, different from rubella, the rubella virus. It's also transmitted by respiratory secretions or droplets. It then enters the respiratory system and travels throughout the body to other organs. It can cause fever, it can cause trouble swallowing, but it does cause one unique symptom called parotitis, which is inflammation of the parotid salivary gland, and sometimes just on one side, as the picture shows. So the parotid salivary gland is the big one in the cheek area, and so you've got a big inflammation right in the cheek area. Now, some of the other symptoms, it can lead to meningitis, it can lead to pancreatitis, and it can even lead to deafness in one ear. Before we developed a vaccine, it was the #1 cause of acquired deafness in humans. And for some individuals, asymptomatic, don't even know that you're a carrier of it. Now, treatment, nothing specific. Most recover on their own and even have long-lasting immunity, but we do have a vaccine. It's in the MMR vaccine, it's the second M. It's measles, mumps, and rubella. Now, the vaccine is approximately 90% effective. So even if you get vaccinated, you still have a chance, a 10% chance of picking it up. But that means you do have to be in contact with someone that has the virus and be the unlucky 10%. So the idea is that if we vaccinate enough individuals, even if you were the unlucky 10% that the vaccine didn't work, if you're not around anyone that has this particular virus, you'd never be exposed to it. It's called herd immunity. Now, they have been recommending just in the last year and a half that adults, before they head off to college, so around 18, to actually get a third booster shot to make sure before they head off to college that they really have the strongest immune system they can have. Because it was just a few years ago that UWL, a couple students went home for spring break and had actually picked up the virus and brought it back with them. So it's around. And the fourth of our Paramyxoviridae family is the respiratory syncytial virus, also called the pneumovirus. It affects the lower respiratory tract, usually just in young children, and it's the #1 cause of fatal respiratory disease in infants and young children. It forms those fused cells, that syncytia, again, it's in its name, in the lungs. And so you're not going to have proper oxygen exchange in the lungs. Now, older children and adults, mild colds, sometimes asymptomatic. It's those infants that have to be worried. Now, it's transmitted by respiratory droplets as well as picked up on contaminated surfaces. And because of that syncytia formation in the lungs, it causes what's known as dyspnea, difficulty breathing, also known as croup. Now, diagnosis, They can look at symptoms. They can also do an immunoassay. They can do an immune system test, and it's a quick test that's going to give you quick results. Now, right now, all treatment is just supportive treatment, giving various types of antivirals to try to slow the virus replication. But right now, just the best prevention is good hygiene to try to decrease the spread of this virus, especially to infants and newborns. Now, our next family of our negative sense single-stranded RNA is the Retroviridae family, and the main virus found in the Retroviridae family is the rabies virus. Now, it's a zoonotic disease of mammals, which means it is primarily found in animals, and we pick it up with direct contact with animals. Now, the main reservoirs, but they're not the only ones. The main reservoirs where it's picked up is infected bats and infected dogs. And it's transmitted by the bite of one of these animals because this virus congregates in the salivary glands. Now, and that means when you get a bite, you have an open wound and you have a larger concentration of the virus from the saliva. Now, my note on the little shape up here, this is a virus that has a bullet shape to it. So it's kind of a unique shape all in itself. Now, the virus attacks the nerve cells, and it attacks all the nerve cells throughout the entire central nervous system. And so the symptoms that are going to appear are going to be neurological symptoms. One unique neurological symptom is hydrophobia. Everyone's like, what? Fear of water? No, you're not going to go run from a puddle. However, it does hurt to swallow water. And so because of that pain with the swallowing of water, animals will stop drinking. And so they call it hydrophobia. It can lead to seizures, disorientation, hallucination, paralysis, and yes, generally by the time you have any symptoms whatsoever, it's going to lead to death. It's an extremely deadly virus. Now, the predominant wildlife reservoirs in our area, our top animal around here that seems to have the highest number of cases of viruses are skunks. But unless you get bitten by a skunk, it's not that common that it's going to spread to humans from them. Now, diagnosing them, again, they're going to look for some of those neurological symptoms. Again, problem is by the time you have symptoms, treatment is too late. So some of the initial neurological symptoms, so before you get to the seizures or anything, although seizures is out there too, but before you get to the hydrophobia, is you could have unexplained anxiety, fear, unexplained fear, twitching, pain with swallowing, hallucinating, seizures, sometimes double vision. Now, the symptoms can appear anywhere between 3 and 8 weeks after a bite site. So it can take a long time before you even see symptoms. And again, by the time you have symptoms, it's too late. Now, one other way they can use for a diagnosis are called nagribodies. When they look at nervous tissue or brain tissue, the virus, when it gets inside the cells, has these dark staining circles in them. Now, if you're diagnosing based on looking at the nagribodies, this is all done post-mortem. So this is done after death, and they want to do a confirmation. Now, treatment. We try to treat, and again, we try to treat right after you think you've been exposed to it. This is treatment right after you've been exposed. This is not treatment after you develop symptoms. By the time you have symptoms, it's too late. So treatment at the site of infection, they're going to clean it. They're going to add an antibody serum right to the wound. Then they're going to give you some antibody, the immunoglobulin, and then they're going to vaccinate you. Now, most of the time you can't vaccinate after you've been exposed. You can't just, oh, well, now I think I have measles, I'll go get the vaccine. However, with rabies, you can. This particular virus is slow, that actually your immune system is faster than it. So if you think you've been exposed to it, you can then get vaccinated for it. Now, they usually give you 4 shots, so it's not just one shot and you're done, and they do it every couple days. It's usually day 0 when you think you've been exposed to it, 3 days later on day 7, day 14. Now, our best bet is, well, let's not get it, and we don't generally give everyone the vaccine because it's pretty rare that you're just going to randomly pick up this particular virus. So instead, we vaccinate pets. So they're the highest chance of having it and then transmitting it to us, because just playing around with them kind of roughhousing with them, they could accidentally bite you and transmit the virus. Now, the third family of our enveloped, non-segmented, negative single-stranded RNA is the Filoviridae family. Now, these generally are going to cause hemorrhagic fevers. Why they get the Filoviridae family is they say that the virus is super long and skinny. So they say it's filamentous. So it's this big, long filament. Sometimes it's long enough it can fold over on itself, curl around itself. And the main viruses that affect humans in this family are the Marburg and the Ebola viruses. And they both cause hemorrhagic fevers. Now, it's unknown as to how it came to infect humans. The thought is, again, maybe bats, but again, they're not really sure where the virus exactly came from. We know it's spread by contaminated body fluids and syringes. We know the virus attacks macrophages, it attacks the liver cells, and it causes uncontrolled bleeding under the skin and from any body opening. It's truly destroying your cells internally. Now, the symptoms can be the diagnostic tool, treatment, trying to replace fluids that are lost, as well as a certain antibody that we've engineered it. It's called a ZMapp antibody. These are not antibodies that animals or humans made. We've engineered these antibodies, and that's just been in the last couple years, and they're having pretty good results because before those ZMapp antibodies, 90% of Ebola victims died, and 25% of Marburg victims died. I'm like, not great odds. Now, luckily, we don't have Marburg and Ebola viruses around here. Most of the cases of them are in Africa. So those that travel to various parts of Africa are going to be more likely to pick it up. People that work with Ebola or Marburg are always going to be in pretty much your whole full hazmat gear. You're going to have your own air supply. You don't want any possible exposure. The highest concentration are going to be these countries on the western coast of Africa. Now, we have had cases of Ebola here in the United States. It's all for people that have traveled. They picked it up somewhere else and brought it back with them. Now, our next group are enveloped, segmented, negative sense, single-stranded RNA. So all of their RNA is in tiny little segments. Now, there are three families, the Orthomyxoviridae, the Arenaviruses, and the Bunyaviruses. And we're going to start with the one most of you are probably familiar with, the Orthomyxoviruses, because it's viruses in this family that cause the flu. And there's lots of flu virus strains. The Bunyaviridae and Arenaviruses, a lot of them are animal viruses. Not all. We'll go through exceptions. And they are spread because of infected animals transmitting them to humans. Now, the Orthomyxoviridae. There are two main types, Orthomyxovirus type A and type B. So when they talk about, I've got flu type A or flu type B, there are two different strains of flus. And then from there, there's a whole lot of strains. Like there's two strains, two groups or two types of flu. And then there's a whole bunch of strains in each of them. Hundreds of type A, hundreds of type B strains of flu. Now, they're primarily spread through airborne transmission. Someone coughing, sneezing, it gets in the air. And once it gets into the body, its main target is the lung cells. These viruses destroy the epithelial lining of your lungs. Now, your epithelial lining of your lungs is your first line of defense because everything you breathe in has the potential of crossing that mucous membrane in the respiratory tract. So when this destroys the epithelial lining, the chance of you getting a secondary bacterial or secondary viral infection is extremely likely. So a lot of times people don't die of the flu, they die of some secondary infection they picked up because the flu destroyed their lung, first line of defense. Now, the symptoms of the flu, fever, malaise, feeling tired, headache, and myalgia or muscle pain. Now, the one symptom that's not up here that everyone seems to catch is vomiting or diarrhea. Those aren't symptoms of the flu, whether you realize or not, because people are like, oh, it's the stomach flu. No, that's not a thing either. The flu is the flu. When people say the stomach flu, they're thinking of a norovirus, but that's not the flu. It's not an influenza virus. Norovirus is an abdominal virus. The flu is the flu. There is no vomiting. So what if you have the flu and you're vomiting? Well, lucky you now have two things. You have the flu, and because you lost that first line of defense, you picked up something else. So if you've got the fever, the malaise, the headache, the muscle pains, and you're vomiting, you're fighting the flu plus something else. So vomiting is not a flu symptom. Now, it is the cytokines, your immune system chemicals, that actually trigger all the symptoms. It means your immune system is trying to work. And because of that, you end up with symptoms of the flu. Now, the structure of the flu. I said this is the group that the RNA is all segmented, and the flu has 8 segments of RNA, and the outside has 2 different glycoproteins. I'm like, I don't care that you know the full name, NAs and HAs. It's just how I refer to them. Now, the NAs and the HAs will form various patterns. This one almost has a pattern of 3-to-1. It's not completely 3-to-1, but it almost has a pattern of 3-to-1. But the genome, the RNA that's in here, varies a lot. And any slight change in the RNA means there's a slight change in the pattern of NAs and HAs, which means it's a completely different strain. So anytime you change any type of sequence with the NAs and the HAs, you have a whole new flu strain. So there's hundreds of sequences, hundreds of flu strains, and they're still changing. Now, different ways we get new flu strains. The first one is the most common. There is some type of recombination, a shuffle of the genes, a mutation that can happen that changes those NAs and HAs that allows us to get a new strain. Now, this changing of the genetics and changing of the existing flu virus is called antigenic drift. And it's antigenic drift that is the number one reason why you need a new flu vaccine every year. When you get a flu vaccine, that is a combination vaccine. It's vaccinating you against several strains of flu viruses. They're tracking, the CDC is tracking, well, what are the strains doing? Do there seem to be more NAs or HAs this year? What are the patterns doing? And they try to make a pretty good guess at getting the right virus in the vaccine. But they're not always right. And then it's changing all the time. So they're constantly looking at these changes. So antigenic drift is why you need a new flu vaccine. Antigenic shift also gives us new flu strains, but it does it differently. It's when you take 2 different viruses, 2 completely different viruses, mix their genes together, you get a new flu strain. So antigenic drift is one flu strain and it shifts or changes or drifts or changes a little bit. Antigenic shift is you get 2 completely different flu strains and their genetics, their RNA mixes together. Now, just to show you the different, antigenic drift, you have kind of a greenish, tealish colored virus goes into a host cell like one of your cells. And then some type of mutation happens, some type of change in the RNA happens, and the virus that leaves your cell is a completely different strain. It's a slightly different color. This is why when you get the flu, this virus that leaves your cells could be slightly different than what you actually picked up because of these mutations. Now, antigenic shift, the fact that you have 2 different strains of flu viruses, they can go into a cell, the viruses shuffle up, recombine their different genomes, and then what leaves is a completely new virus. And again, your book likes to go red plus blue equals purple. And so 2 different viruses, now you have a completely new virus. So this is why you need a new flu vaccine this year. This is how we get really new, completely different flus. Now, the interesting part about this is that antigenic shift can happen quite often, but the interesting part, we seem to have outbreaks of this about every 10 years. The swine flu was 10 years ago, and well, we got a new flu now. The new flu we have not the new flu, but the new coronavirus, it could have developed from something similar to antigenic shift. an animal virus and a human flu virus mixed together, and we've got a brand new virus. Now, diagnosing the flu, a lot of times they can look for signs and symptoms. They're very characteristic signs and symptoms. Treatment, we do have a few antivirals. Oseltamivir or zanamivir, more commonly known as Tamiflu, can be given if you're given in the first 48 hours of infection. They block the virus from being released from infected cells, so it shortens the illness because less cells are going to get infected. Prevention, though, is that we can get rid of infected birds or other infected animals so that we don't get these new flu strains, and get vaccinated. Now, it's called a multivalent vaccine because it is a combination vaccine against multiple strains of flus, and it's depending on the season. Some are more effective than others, but even if you get the flu and you were vaccinated, you generally have milder symptoms because all flus still have some similar characteristics from each other. They're all still genetically a flu. They're still an influenza virus. And you would still have some antibodies that can recognize some parts. So you normally have a milder case of the flu if you get vaccinated. Now, our next group away from the flus are the Bunyaviruses. Now, Bunyaviruses a lot of times are zoonotic pathogens, so animal viruses that are spread through biting arthropods, so insects. So it's a virus found in an animal, some type of fly, mosquito, tick bites an animal, then bites you, and you pick it up. Now, symptoms are usually mild. However, there can be some more severe cases like Rift Valley fever causing a really extreme fever, California encephalitis, and our very own La Crosse encephalitis is a Bunyavirus. One unique virus that's in the Bunyavirus group, it's spread by animals, not arthropods. It's called the hantavirus. We pick it up from inhaled dried mouse urine or feces. Now the virus, when you inhale it, gets into the lungs, it infects the lungs, destroys lung tissue, and it can cause a fatal pneumonia. Now, there are cases around here, but extremely rare. So there are cases found more in different parts of the country, rare around here. Your best bet is to not pick up a Bunyavirus. If it's spread by arthropods, limit your contact with various biting insects, wearing bug spray. For hantavirus, rodent control so you don't have mouse urine or feces in your house or wherever you are. The Arenavirus group are ones that can cause hemorrhagic fevers. They're spread by rodents in Africa and South America, and they can cause severe bleeding underneath the skin. So that's the hemorrhagic fever. So it's internal bleeding as well as a fever. Now, another disease disorder they can cause is called the lymphocytic choriomeningitis. And although its name does have meningitis in it, it doesn't actually cause meningitis. They at one point thought it did, and the name just stuck. Instead, it typically causes flu-like symptoms. It's spread by aerosols, contaminated food sources, and one other virus that can spread it or can cause choriomeningitis is the Lassa fever virus that's spread by bodily fluids. Now, treatment for any of the arenaviruses are all supportive care. We don't have any specific treatments. The best prevention is limit yourself with infected rodents or any of their urine or fecal matter, and good hygiene, wearing proper PPE, things like that are going to decrease the chance of picking it up. Luckily, most of the viruses in here, not found here in the United States, very common, but if you're ever traveling, you are putting yourself at risk for some very unique viruses that are out there. One type of arenavirus, though, that's not a zoonotic virus, it's not spread by any kind of animals, is a hepatitis D, because we still had to get to hepatitis D. It's our last hepatitis, and it's transmitted by bodily fluids, so sexual activity, contaminated needles. But the interesting part about hepatitis D is that it requires hepatitis B to reproduce. It steals the hepatitis B surface proteins that allow it to attach to the liver and cause inflammation of the liver cells. Now, if you have hepatitis D and it can get into the liver cells because you also have hepatitis B, it does play a role in triggering liver cancer, but it is a severe inflammation of the liver. So a lot of times when you have a hepatitis D infection with your hepatitis B infection, it usually causes permanent liver damage. Luckily, although we don't have a vaccine specifically for hepatitis D, we do have a vaccine for hepatitis B. And if you vaccinate for one virus, it actually stops the other virus. Because this virus requires hepatitis B, if you eliminate it, it can't get inside the liver cells. So by vaccinating for one thing, you end up vaccinating for two things. Now, the last virus group is just one single little group, and it's a unique group all in itself. They're naked, they don't have an envelope, segmented, double-stranded RNA. Now, it's the Retroviridae group, and there's only one main virus in there. They're the Rotaviruses. Now, they get the name Rota because they say they look like rotating wheels. So that's where they get their name, is what they look like. And it was the leading cause of infantile gastroenteritis, mostly in developing countries, due to the diarrhea. It's just losing the fluids and electrolytes. Now, it was the leading cause of infantile gastroenteritis. We now do have a vaccine. It just depends on how many individuals are vaccinating. It is spread fecal oral, and well, kids are dirty, so they are usually the ones that are picking this up. They said that in different countries, almost 100% of children within the first few years of your life had picked up a rotavirus at some point. Now, it will leave on its own. It will leave with the diarrhea. So the main treatment is fluids and electrolytes. But if you can't replace fluids and electrolytes, it could be a deadly symptom. Now, best prevention, good hygiene, you spread fecal oral, and sewage treatment so that we don't have infected sewage anywhere to become an infection source. Now, the vaccine we have, it's relatively new on the market, and it's about 75% effective, which again, it's still pretty good odds, especially if you can get enough people vaccinated that no one's spreading it. However, it is still causing deaths in developing countries if they don't have access to the vaccine and they can't replace the fluids and electrolytes. Now, this is just a nice summary of all the RNA families that this PowerPoint goes over. So from the picorno, those tiny little viruses, and it goes through various types of groups of viruses in there. So we've got lots of family, lots of viruses in those. But otherwise, we've gotten through all of the RNA viruses. So thanks for sticking with me.